Dear students, I am Dr. Poonam Agrawal. Today I'll be talking to you about gene expression regulation. Gene expression regulation in prokaryotes. Gene expression means synthesis of protein from the gene. Now, in this context, I would like to tell you that in prokaryote, it is the level of transcription at which the gene expression is regulated. And when we talk about eukaryote, we have multiple levels of gene expression regulation, like at the level of transcription, post-transcriptional modification, then translational, then post-translational modification. Multiple places we have regulation of the gene expression in eukaryote. But in prokaryote, we have, importantly, having the regulation at the step of transcription, where the gene is forming RNA. In this context, Francis Jacob and Jacques Monod, they proposed a model which was known as Lac-Operon model in 1961. This model explains how the prokaryotic gene expression is regulated. Importantly, this model explained the molecular basis of synthesis of lactose metabolizing enzyme in presence of lactose and simultaneous absence of glucose. So, E. coli, the organism which uses lactose, produces lactose metabolizing enzyme in presence of lactose and absence of glucose, and the molecular basis of this was explained by Jacob and Monod in 1961, for which they were awarded Nobel Prize in 1965, and that model is called Lac-Operon model. First of all, you should be familiar with the terminology operon. Operon is an umbrella terminology. It is all the elements on the DNA which are involved in gene expression. It's a unit of gene expression. And one operon consists of multiple components: the structural gene, the operator area, the promoter area, the control elements, and the inhibitory genes. So these components together is called operon. So in Lac-Operon, we are talking about these elements which are involved in regulation of lactose metabolizing gene expression. So in this model, they explained that the DNA of E. coli, this is DNA of E. coli, is having following elements on it. This is the structural gene, 1, 2, 3. There are three enzymes which are needed for lactose metabolism, and the gene for them is ZYE. Z is the gene for beta galactosidase, which acts on lactose to convert it to glucose and galactose. Gamma is the gene which is responsible for production of protein permease, which is responsible for permeation of lactose and galactose in the E. coli. And this A is the gene for enzyme thiogalactoside acetylase, which is needed at one of the step of lactose metabolism. So these are structural genes, and these scientists, they said that upstream to this structural gene there are operator area, and further upstream we have promoter area. Further upstream we have control elements, which is called CAMP binding element, and further upstream we have the inhibitory gene, which is responsible for making of inhibitory protein, which is called repressor protein. This repressor protein is by default getting produced. This is constitutive gene always expressed inhibitory gene is constitutive gene, which is always expressed. It produces repressor protein which is having tendency to bind the operator area, and thus the operator area by default is blocked by repressor protein. You know, promoter is the area where RNA polymerase enzyme binds. So when RNA polymerase enzyme binds here at promoter area, it is not able to read the gene ZYA and it's not able to make the polycystonic mRNA just because the operator area is blocked by the repressor protein. So these scientists, they proposed the model and they said that for making the operator free, see, if the RNA polymerase enzyme need to read the gene and make the RNA, the operator area should be freed of this repressor protein. To make the operator free, they said the presence of lactose is required, because this lactose is making its isomer allolactose, and that allolactose is having a strong tendency to bind these repressor protein. This allolactose tend to bind these repressor protein and destroy them. So in presence of lactose, when we have allolactose, which is isomeric form of lactose, small amount of lactose is converted to allolactose, and that allolactose binds with the repressor protein and prevents its binding at the operator area. This makes the operator free, and now RNA polymerase can move ahead and can scan the gene, can read the gene, can transcribe the gene, and this polycystonic mRNA is getting produced, which then is getting translated, producing these enzymes needed for metabolism of lactose. So this could explain why the presence of lactose is important for gene expression. At the same time, they explained that how the lack of glucose is essential. Glucose absence is essential for adequate gene expression, for considerable gene expression, which can produce considerable enzyme needed for metabolism. So for that, they explained this glucose absence activates adenylyl cyclase, which in turn produces cyclic AMP. There is a protein in lactobacillus which is called CAP, that is catabolite. By default, this protein is there, catabolite activator protein. Now when this cyclic AMP binds with this CAP over here, it becomes CAP-CAM complex. This is cyclic AMP. This is binding the catabolite activator protein. This catabolite activator protein also is known as, at times known as CRP, CAMP response protein. Cyclic AMP response protein. So this CAP-CAMP complex, if produced, when it will be produced in lack of glucose. So when this CAP-CAM complex is produced, this complex will go and bind the CAP-CAM binding area. It will bind the CAP-CAM binding area, and this will act as a positive regulator. This will enhance the transcription. It will induce the transcription 50-fold. So they named this complex as positive allosteric modifier of Lac-Operon model. So if this complex is formed, the gene expression increases by 50-fold. So to this particular structure, they named it as positive allosteric modifier, for making of which you need to have absence of glucose. So this could explain why the presence of lactose and absence of glucose is important for adequate gene expression, which can metabolize the lactose. By default, the lactobacillus gene ZYA, they are under repression by repressor protein. For derepression or for induction, the lactose presence and glucose absence is required. This is Lac-Operon model, this you should know. And this was the first model, primitive model, which was talking regarding the regulation of gene expression. right? So in next class, I'll be discussing about the prokaryotic gene regulation. So now you should understand that this lactose was said to be inducer by them. They said this is inducer because if it is present, it forms your allolactose, and that induces the whole process. And positive allosteric modifier, I said it's CAP-CAM protein. To repressor protein, they said they named it as negative allosteric modifier. Negative allosteric modifier. Negative allosteric modifier was repressor protein. Though today's date, this allosteric modifier terminology is not very correct when we are talking about genetics. When we are talking about the elements which are factors which are binding the DNA. If the factors are binding the protein, the enzymes, and modulating its activity, we call it as allosteric modifier. But in that era, when the study on enzyme was going on, and that time this allosteric terminology was used for enzyme regulation, right? So they proposed this terminology allosteric for these factors, which is not very correct. It's actually a misnomer, but in their honor, these terminologies are still retained. So this is, in a nutshell, is a Lac-Operon model which you should be clear of. Thank you very much.